Biological databases have been invaluable for managing these data and for making them accessible. Depending on the data that they contain, the databases fulfill different functions. However, one of the major problems is that each of these database resources contains a different subset of biological knowledge. Although each database can answer questions in its domain, it cannot help with questions that span domain boundaries. For example, the FlyBase database does not know about orthology relationships. The Institute for Genomic Research, TIGR, database can help with orthology, but not with map position, and the UCSC database knows nothing about the fly. As this example shows, researchers must become adept at database surfing to answer many reasonable questions. Imagine, life would be much simpler if there was a single biological database. But this would be a poor solution. The diverse databases reflect the expertise and interests of the groups that maintain them. A single database would reflect a series of compromises that would ultimately impoverish the information resources that are available to the scientific community. A better solution would be to maintain the scientific and political independence of the databases, but allow the information that they contain to be easily integrated to enable cross-database queries. There are many integration challenges. One of the most difficult is the one that might seem the most minor. How do you assign and maintain the correct names of biological objects across databases? For example, consider the DNA damage checkpoint pathway gene that is named RAD24 in Saccharomyces cerevisiae, or budding yeast. Saccharomyces pomorficin yeast also has a gene named RAD24 that is involved in the checkpoint pathway. But it is not the orthologue of the Saccharomyces cerevisiae RAD24. Instead, the correct Saccharomyces pomorthologue is RAD17, which is not to be confused with the similarly named RAD17 gene in Saccharomyces cerevisiae. Meanwhile, the human checkpoint pathway genes are sometimes named after the Saccharomyces cerevisiae orthologues, sometimes after the Saccharomyces pom orthologues, and sometimes have independently derived names. A more subtle problem is the clash of concepts as users move from one database to another. For example, the use of the term pseudogene, genomic variant, and even the concept of the gene itself can mean radically different things to different research communities. Some researchers treat the gene as the transcriptional unit itself, whereas others extend this definition to include up and downstream regulatory elements, and still others use the classical definitions of cystrin and genetic complementation. There are also technical challenges. The various biological databases use different DBMS, and none provide a standard way of accessing the data. Even more challenging is the issue of updates. Biological databases are always changing, so integration must be an ongoing task. There are main ways in integration of biological databases can be achieved. First, link integration. Link integration has been by far the most successful, because it lends itself to the haphazard nature of the web. In the familiar and ubiquitous version of this technique, researchers begin their query with one data source, and then follow hypertext links to related information in other data sources. Second, a variation on link integration is embodied in the popular sequence retrieval system, which is a keyword indexing and search system for biological databases. Sequence retrieval system is more sophisticated than general web-based search tools, for example, Google, because it recognizes the existence of structured fields in source databases and allows maintainers to explicitly relate a field in one database to a differently named field in another. Unfortunately, link integration is problematic on several fronts. First, it is extraordinarily vulnerable to naming clashes and ambiguities. For example, a naive researcher who tries to navigate through the RAD24 gene confusion using SRS or direct database-to-database linking might wander into the wrong gene family. Second, there are update issues. An outgoing web link represents a leap of faith that the page at the other end of the link is still valid, or if it exists at all. For example, if the curators of the Saccharomyces cerevisiae database withdraw or rename RAD24, they have no way of informing all the databases that point to the RAD24 page that an update is required. Third, view integration. View integration leaves the information in its source databases, but builds an environment around the databases that makes them all seem to be part of one large system. Despite the appeal of this approach, the system has failed to catch on with the community. Fourth, data warehousing. Data warehousing can be broadly described as bringing all the data under one roof in a single database. The first step in data warehousing is to develop a unified data model that can accommodate all the information that is contained in the various source databases. The next step is to develop a series of software programs that will fetch the data from the source databases, transform them to match the unified data model, and then load them into the warehouse. The warehouse can then be used as a one-stop shop for answering any of the questions that the source databases can handle, as well as those that require integrated knowledge that the individual sources do not have. However, it is more difficult to create a data warehouse than it might sound. The single biggest issue is keeping the data warehouse up to date. New information is being continually added to the source databases, which means that the new data must be re-imported into the warehouse in a timely fashion, or the warehouse will go out of date. To make matters worse, database designs do not stand still. Their maintainers are continually tinkering with the data model by adding new data types, changing fields and nomenclature, and changing the relationships among data types. This constant churn means that dump, transform, and load software that have been written for one version of a database will not necessarily work with a later version. Fifth, web services. An important variant of link-level integration is the concept of the web service. In this view, the heterogeneous collection of loosely linked data resources on the web is turned on its head and becomes a world of services that are linked by service names and definitions. In such a world, GenBank is no longer a database from which users retrieve sequence entries. Instead, it is a service that transforms sequence accession numbers into GenBank flat files. This concept might seem a minor point, but it allows users to establish a common framework that encompasses, for example, the BLAST search engine, which transforms a search sequence into a series of similarity hit records on a selected query database. Finally, establishment of globally unique identifiers. Recall the issues that the same biological object might have multiple names, and the same name might denote multiple objects. One approach is to designate an authoritative names commission to manage the definitive list of such names. SU effort is currently being handled by the Hugo Gene Nomenclature Committee, or HGNC in short.